Okay, so first of all, thanks Jessica for the introduction and the entire committee for giving me this opportunity. And this has been a wonderful series of talks, and I really hope I can live up to the standard that has been established. So, and for my talk today, I'll basically talk about sexual reproduction. The theme of the talk is going to be on that part. And to introduce you to that concept, basically, all of us are aware of this process, sexual reproduction, which basically starts when you have two opposite sex gametes, in this case, a sperm and an egg. They fuse together and you get a zygote. The zygote then matures into a diploid organism where you have germ cells, and a round of meiosis takes place, which again produces these gametes, and the cycle continues. Now, meiosis is an important step in this because it basically mixes up the two parental genomes and it generates diversity. It filters all the harmful mutations in that, and which basically allows the adaptations. So these are all the benefits of having sexual reproduction. But in addition to these benefits, this can also be a very energy costly process, and sometimes in the wild, finding a suitable partner can be challenging. So there are certain disadvantages along with it. So it's not a surprise that some organisms have basically adopted alternative forms of this. So the most commonly known form is parthenogenesis, where you have a female producing diploid eggs, which basically develop into an entire organism, and there is no requirement of any partner in this process. And then the other alternate approach that some organisms take are gynogenesis and hybridogenesis. And these are collectively called as sexual parasitism. And the reason for that is because, for example, in gynogenesis, the female produces diploid eggs, but it needs the sperm just for the induction so that the progeny can be can develop. Now, so basically sperm never contributes to the progeny in any way. In case of hybridogenesis, it starts very similar to what happens in sexual reproduction. You have gametes from opposite sex partners, and then the organism is formed, but this organism then gets rid of one of the parental genome and passes entirely only one of the genomes to the next round. And so this guy here never gets to contribute anything to the progeny line in any way. And that's why it's called sexual parasitism, because it feels like an exploitation. Now, again, but these are alternate approaches. So it happens only in a very limited number of organisms. And so far it has been observed in insects. This is the stick insect here, or in fishes, mostly in amphibians and reptiles. Today, what I'm going to try and show you is that this is something that can also happen, or at least happens in fungal kingdom, but it has not been seen previously. And the fungus that I'm going to use is Cryptococcus neoformans. Now, most of you are well familiar with this fungus. And so this is a fungus which is present everywhere in the environment. It infects humans in the form of spores or desiccated yeasts. When the infection starts at the lungs, but it can disseminate into the rest of the body, causing cryptococcal meningitis, which is the deadly part of this infection. Now, these infections result in more than 180,000 deaths across worldwide, which basically accounts for 15% of HIV/AIDS related death. As a fungal system, this fungi basically exists mostly in the yeast stages. So, and it divides by budding, where it basically throws off bud, which matures into a new daughter cell, and the cycle continues. But it also has a sexual cycle, which basically comes into picture when you have the two opposite sexes here, which are known as mat A and mat alpha. Now these are the two mat A and mat alpha I'll be repeatedly using in my presentation. Now these two cells of opposite sex, when they come together, conditions are favorable, they fuse with each other, they form this zygote, which then basically takes a different turn develops into a filament. This filament then matures into a basidium, and only when the basidium is formed, the two parental nuclei fuse with each other, they go through meiosis and produce these spore chains. Now, like I said, my talk is going to be focused on sexual reproduction. So this is the cycle that I'll be talking about. And so to give you a more idea of how we do this in the lab or how we study it, we basically just take two strains or a mix of cells of both these sexes, mix them in a tube, put it on the MS media, and then incubate them. And you can then slowly see, like in a couple of weeks, these hyphae develop, and then you start seeing these basidia, which come out in a time of about 2 weeks. So the story that I'm going to talk to you today basically started a couple of years back when we generated this strain chromosome shuffle, which had about 8 different chromosomes variation compared to the wild type H99 strain. Now this is H99 is the lab strain that is used probably by most people who work on neoformans. So now there was no difference in the gene content of these two strains, only the structural variation in terms of chromosomes. And these structural variations were generated by CRISPR against the centromeric regions. So, and in the rest of the talk, I will basically refer to this as wild type and this as chromosome shuffle, just not going to the names and details. This story was published early last year. But what the main gist of this was that when you take this chromosome shuffle strain and cross it with a MAT-A strain, which had the same karyotype as wild type, it leads to severe defect in sporulation. So it mates well, everything else goes fine. It forms these robust filaments. But unlike the wild type strain, where you have these spore chains, both in light microscopy and in scanning electron microscopy, this cross does not give rise to much sporulation even. So you have basically see this bald basidia phenotype in this case. Now this was in a matter of 2 weeks. But when we looked at these crosses again at a later time point in about 5 weeks, we started seeing a few basidia which had nice decorated 4 long spore chains on them. So shown, and this was roughly about 1% frequency. So most of the basidia are still bald. They don't have much spores on them, but there are a few that do. So that got our attention, and we started looking at these in more detail. And we did that by dissecting and analyzing one basidia at a time. So what happens is when you take out these basidia and dissect out individual spores from them, you get both parents in the progeny. So you have alpha parents and A parents, and that's how it should happen because they go through meiosis, mixing the two parental genomes. So basically this part here, but if you look at the same thing in case of chromosome shuffle strain, all the progeny that germinates inherits only one of the parental mating type, either alpha or A. And doesn't matter what the germination frequency is. And so this was the very peculiar pattern that we observed at this stage. Now, one thing to note here is that the mitochondria in both these cases is always inherited from MAT-A parent. And that's something that's well established in the field by now. So this analysis basically raised a question whether this is a biased inheritance of only the mating type, that there is some kind of effect that's happening as a result of that, or its entire nucleus. So to dive into that, we put basically colors in these two parental strains. So we tag the nucleus in the MAT-A strain with M cherry and the nucleus in the MAT alpha strain with GFP. So as they progress, go through the mating, you will see these yellow orangish nuclei after they are fused because they are in a common cytoplasm. And then you will see these basidia and spore chains, which will have both red and green fluorescence in them. Now we hypothesize at this stage that if only one of the nucleus is making those spore chains that we had observed, what we will see is instead of this yellow-orange color in the spore chains, we will have only green or only red, as you can see here. So once we established this assay, we started looking at the spore chains in both the cases. So if you look at it in case of wild type, you have most of the basidia will have the red fluorescence and the green fluorescence, and this is how it appears. So basically both parents are involved in sporulation. But then there was about 1% of population which had only one of the fluorescence color in them. So for somehow the second parent nucleus was absent from here. We then did the same analysis with the chromosome shuffle strain, and like similar, about 1% of basidia had spore chains, and they were either green or red, but not orange. And if you look at the basidia which are both red and green, they seem to be stuck in some stage, but fused with each other, and they don't form any spore chains on them. So that was pretty convincing that there is something happening here where only one of the parent is basically forming the spore chains. But this was all done with the lab strains and stuff. So we wanted to know whether this can happen with natural isolates. And to do that, we selected two of the natural isolates. One is BT63A and one is IUM96. This was in an environmental, this is the clinical one. And so this is a tree which basically shows all the different isolates of crypto that, not all, but most of them, and the different lineages. So BT63A is very different from H99, which is the parental strain for 135. And I'll be talking only about BT63A today in the interest of time. So now, in addition to having this difference in terms of genetic divergence, which is about 0.5 for BT63, it also has this and one additional reciprocal translocation, which basically makes one step harder for BT63 to cross with 135 strain. So we thought this will work out in a similar fashion as we had expected. And that's exactly what happened. So when you do it for wild type H99, you see these nice spore chains popping up. And then when you dissect them, the spore chains, you have the progeny that is a mix of A, alpha, and sometimes, or rather most of the times, aneuploids in this particular case. And then if you do it for 135, you see that all the progeny is coming from inherited basically only the MAT alpha allele, but there is no MAT A in this case. Now, again, in mitochondria is always inherited from MAT A, which is consistent. One other thing that I would want to get your attention to is the germination rate. Now, the germination rate here is pretty low, but it's pretty high in this case. And I will come to this point at the end of my talk. So with these progeny in hand, we also wanted to look at them in more detail. So one thing that this genetic divergence of about 0.5% allowed us to do was to sequence the progeny and see if there is any kind of recombination or mixing up of two parental genomes happening at any stage. So the way you basically do is you, in a case of a normal cross, you have this diploid zygote, which forms these 4 spore chains, and they undergo meiotic recombination. So their genomes are basically have a mix of both the parents. So you can sequence them using Illumina, map them back to the reference, and you see these signatures where a part of it, the genome is coming from one parent, but then there is another part which is coming from other parent. And you can detect it by looking at those. So we did the same analysis, and we see that in case of wild type progeny, if BT63 is yellow, H99 is green, each of the progeny has mixed genome from both the parents, suggesting the evidence that there is meiotic recombination happening there between the two parents. Now, in this case, however, always in all the progeny that we had, the genome entirely belonged to 135. There was no trace of BT63 genome any of these progeny, suggesting that the two genomes never mix up with each other. So probably one of the parental nuclei is left behind at a much earlier stage before it even develops into a basidium. And that raised the question, when does this happen? So when do the two parental nuclei separate? So to understand that, we imaged long hyphae which had the spores on top of them. So in this case, if you do this for wild type, you have these spore chains which have both red and green fluorescence, and you can track the location of the nucleus in these hyphae, and they basically have both red and green fluorescence throughout the length of it. If you do the same thing for the chromosome shuffle cross, and in this case, I'm showing you one basidia which had red spore chains on them. And if you trace it down, it has red fluorescence here, but after this branch, it has both red and green fluorescence. So something probably happens during this branching event that pushes one of the nucleus into a separate branch, which then undergoes this entire cycle and forms these spore chains. So it suggests that these hyphal branch point are one of the ways this entire process can take place. So now, one thing that is important to note here is that this nucleus is a haploid nucleus at this stage. So to make these spore chains, it has to basically go through meiosis. That means we need to look into that as well. So by now, I have showed you that you have these two parental nuclei, they fuse, but they don't go through the normal process of basidium. Rather, one of them is lost, and the other one somehow duplicates and then forms these spore chains. So possibly there is an endoreplication kind of mechanism that takes place, and there has to be meiosis involved with these four spore chains. Now, we don't have direct assay at this point to study endoreplication, but we did look at meiosis. And the way you look at meiosis is basically by studying the genes involved in this recombination pathway. So in the meiosis, basically two parental genomes, they cross with each other, they go through this recombination step. And this recombination step is controlled by multiple genes. Two of them are SPO11 and DMC1. So if you basically knock out one of these genes, what you will have is a failure of meiotic recombination. And as a result, you probably will not see any spore chains in there. And so in our case, we deleted DMC1. And if you do that, so if you look at a wild type scenario, you have nice spore chains forming. If you delete DMC1 in both the parents, there's still a small percent of basidia that bypasses the requirement that forms these spores, but majority of them is basically bald basidia. Now, for the chromosome shuffle strain, this was a bit tricky because there is only a small percent of sporulation events to begin with. And when we looked at it in more detail and did some analysis, we found that this gets further reduced when you have both the parents missing DMC1 in them. So this is shown here in graph where you have about 1% for wild type and DMC1 mutants. If you have deleted both, it gets reduced dramatically. But if you delete only in one of the parent, it is an intermediate phenotype in all the cases. We also did an assay for microscopy with this, where we deleted DMC1 in one of the parents, but not in the other. So that will basically show us that in the basidia which inherited this particular parent nucleus, they will stay bald, they will not form any spore chains, and that's what you basically see. But the basidia that get 135 in them, they form these spore chains around, suggesting that DMC1, or rather meiosis, is an important step that is required for this process. So in overall total summary of what I have told you today, you have in this process, you have these two cells of opposite sex. They come together, form this zygote, form this filament. And unlike the bisexual reproduction where the two parental genomes fuse, in this case, one of the nucleus is lost, The other one replicates its genome, probably via endoreplication, then goes through meiosis and forms these spore chains. Now, we call it pseudosexual because it requires this first step of sexual reproduction, which is mating, but then it doesn't produce the recombinant progeny, rather clonal progeny. And if you look at the steps involved, it is pretty similar to what happens in animals during hybridogenesis, where you have the two parents coming together, but one of them is removed just before the gamete production. So what's the implication of this? So, so far, I showed you that this happens, but it happens only in about 1 to 2% of population. And if you look at the implications of this, it probably doesn't matter to Cryptococcus, at least in the lab conditions, because you always use strains which are compatible with each other. They go through this successful bisexual reproduction, producing recombinant spore genes. And this can be ignored very well. But that's not how it probably works in nature. In nature, the cell will come across all kind of opposite sex partners where it will not be compatible, either because they are highly genetically diverse or they have genome structure variation, or probably both. And in this case, like I told you for the BT63 case, you will have nice spore chains in them, but they will not germinate very well, and that can be severely defective. So in those cases, pseudosexual reproduction becomes a mode of survival, and so does in case where there are no spore chains at all. So while it doesn't give you the benefit, long-term benefits of sexual reproduction, this process can basically aid in survival and then allow spreading of Cryptococcus cells to get more get basically introduced to the more mating partners and then undergo successful bisexual reproduction at the end. So in summary, I told you that Cryptococcus exhibits pseudosexual reproduction after mating in a small population. The uniparental progeny that comes is identical to one of them, and it starts at some point during the hyphal branching. And this process requires meiosis. So this is indeed like everything else is similar to sexual reproduction except the clonal progeny production. With that, I'd like to thank my lab members, Joe here and Sheng, who did all the spore dissections that you saw in those tables. And this story is now on bioRxiv if you are interested in looking at more details, and the funding and support from Duke MGM and NIH. And thank you so much for this opportunity again.
Thank you very much, Vikas. That was a wonderful talk. So we will take please enter questions into the chat and we'll take them after both talks. And now we'll transition to Kyla.
Okay, well, can everyone hear me okay? Good, okay. So I'd like to start by thanking Liz and Jessica for putting on such a fantastic seminar series this year. I've really enjoyed this, and I'm honored to be included as a participant. So I'm excited to tell you about some work that I've been doing in the Round Lab, in which we've identified an interesting mutually beneficial interaction between adaptive immune responses within the gut and a commensal fungus. So of course, you all likely know that the large and complex microbial community that resides within the gut serves many important roles in shaping host health and disease. And I, of course, study the fungi. So fungi are often overlooked members of this microbial community, but it turns out are very important for shaping host health. So despite them making just a small proportion of the total microbial community, emerging research has really established that they have an outsized role in impacting host health and disease. And this community is dominated by yeast species, primarily in the Saccharomyces Candida and Malasthesia genera. And research has demonstrated that these organisms are potentially pathogenic. So of course, most of you probably recognize that a number of these fungal species are potentially pathogenic and can cause life-threatening disseminated infections if allowed to escape through mucosal niche. These are primarily fungi in the Candida genus. But more recent work has established that immune responses induced by fungi within the gut can have pathogenic consequences for a number of inflammatory diseases. This includes allergic airway disease, certain types of cancer, and there's now a growing list of specific fungi that have been shown to exacerbate inflammatory bowel disease. Yet we know that fungi, these fungi are not pathogenic in most people under most circumstances. And really recent research suggests that they may even be beneficial. Yet we know very little about what forces constrain these fungi to a commensal state within the gut to maintain homeostasis. Research on the bacterial members of this intestinal community have demonstrated that adaptive immune responses are vital in maintaining homeostasis with the microbiota. So these responses are, immune responses are orchestrated by T and B lymphocytes that reside in intestinal immune tissue, which are constantly stimulated by microbial antigens. One of the resulting responses that comes from this stimulation is the production of IgA antibodies. These IgA antibodies are secreted in large quantities into the intestinal lumen, even under homeostatic conditions. And there they target and sculpt the microbiota. So they've been shown to be very important in sculpting the community composition and function, in addition to directly regulating the biology of particular commensal bacteria. Yet there's a reason why I tried to make this figure show only bacterial shaped microbes. And that's because when I started my postdoc, there had been no sort of exploration of what these antibody responses, how they interact with fungi, and what impact they may have on fungal host interactions within the gut. And so I started this project with just two very simple questions, and that is, are fungi even targeted by these immune responses within the gut? And if so, what are those immune responses doing to shape fungal biology and interactions with their host? And so to address whether fungi are targeted by adaptive immune responses, particularly in humans, we took a targeted approach in which we took the four of the most abundant members of the intestinal fungal community and asked whether their humans harbored antibodies that react against them. So we took cultures of Candida albicans, Candida tropicalis, Saccharomyces cerevisiae, and Candida glabrata, and incubated them with cleared fecal homogenate from 70 human volunteers, and quantified antibody binding using flow cytometry. And so what we found was that the majority of these human samples had fungal reactive antibodies that targeted at least one of these fungal species. And as expected, the IgA isotype of the antibodies was the most dominated this response. So you can see that here in these black bars. But we found that this response was not just sort of generally reactive against all fungi, and it appeared to be dependent on the fungal species. And specifically, we found that IgA was most reactive to the Candida species, and primarily Candida albicans, and significantly less reactive to Saccharomyces cerevisiae. And so we hypothesized that this reflected a differing capacity of different fungal species to induce antibody responses during colonization. And to test this directly, we moved to mouse models. And so what we did is we took germ-free mice that lack all other microbes and monocolonized them with either Candida albicans or Saccharomyces cerevisiae. And we used strains of these fungi that express fluorescent markers that allowed us to quantify IgA targeting of these fungi in the feces of mice over time using flow cytometry. And so from this experiment, we observed a striking difference between Candida and Saccharomyces. We found that over the course of this 30-day colonization, that the Candida fecal population becomes highly targeted by IgA. You can see here quantified here, an example flow plot here. But in contrast, Saccharomyces cerevisiae never becomes significantly bound by IgA, despite Saccharomyces colonizing these mice at a relatively high level and being the only microbe present within their gut. We found that this IgA targeting of Candida was also associated with increased total IgA, particularly in the small intestine, in addition to increased colon IgA plasma cells and increased germinal center B cells in the Peyer's patches. In contrast, the Saccharomyces colonized mice looked immunologically very similar to germ-free mice. And so together we concluded that Candida albicans, but not Saccharomyces cerevisiae, promotes a specific adaptive immune response during intestinal colonization. And so I focused on Candida to ask what impact these immune responses have on the host fungal interface. And so I first asked what impact do these adaptive immune responses have on the Candida biology during colonization of the gut, and specifically the genes that Candida expresses. And so I again set up this pretty simple experiment in which we took germ-free wild type and immune deficient rag mice that lack adaptive immune responses and monocolonized them with the same strain of Candida and performed RNA-seq on the intestinal Candida populations. And what we observed was a striking effect of adaptive immunity on the Candida transcriptional profile. We found that up to 25% of all of the Candida genes were significantly differentially regulated between these mice. And we were intrigued to note that the genes that seem to be, that were most significantly upregulated in the immune deficient rag mice are really well-known virulence factors that are associated with this famous morphological transition that Candida can undergo. This is the transition from yeast to hyphae. And so many of you are in the audience are probably very well aware that Candida can transform from the single cell budded yeast form to this elongated hyphal cell form. And this is really important for the pathogenic potential of Candida, as these hyphae have increased adhesion and invasion abilities, making it the more pathogenic cell form. And so we asked directly whether this yeast to hyphal transition or these yeast and hyphae are different in wild type or rag mice. First, by comparing our RNA-seq data to a list of genes known to be upregulated during hyphal formation. And so from this gene set enrichment analysis, we found that indeed there was a significant enrichment in hyphal genes in the Candida colonizing rag mice compared to wild type mice. And we also simply looked at the morphology of Candida during colonization of these mice. And consistent with these results, we found that the rag mice, the Candida population with rag mice, was significantly more hyphal than the Candida colonizing these wild type mice, suggesting that adaptive immune responses constrained this fungus to a more yeast state. We asked specifically what sort of immune responses were required for this suppression of hyphae, and found that in B cell deficient MUMT knockout mice, that we also observed a significant increase in the proportion of Candida hyphae in the gut of these mice compared to wild type mice. And so, of course, B cells are the cell types that are responsible for producing antibodies. And so we next asked whether IgA responses may be having a role in a selection against hyphae. And so we set up an experiment very similar to the one I showed you prior, where we monocolonized mice with GFP expressing Candida albicans and used flow cytometry to quantify IgA binding of the fecal Candida. But instead of a regular flow cytometer, we got to use this really neat imaging flow cytometer in which we could actually take a peek of the morphology of the Candida cells in both the IgA positive and negative populations. And so using this technology, we were able to observe that in the IgA positive population, that the Candida are almost all hyphae. So all hyphae, you can see here from the examples of IgA positive Candida here. There's clearly elongated hyphal cells with the yellow IgA shown here. And so this was great. It suggests that that adaptive immune responses target specifically these hyphae within the gut to suppress them during colonization. But we wanted to ask what specific molecules may be targeted by IgA within the gut. And so we took a number of approaches, but one of the first ones we did was a genetic approach in which we performed a genetic screen using two available Candida mutant collections, the Noble collection and the Homan collection. And we screened these collections in vitro for IgA binding using IgA from mice that had been monocolonized with Candida. And we quantified IgA binding by flow cytometry. And what we found was that the mutants that had the most significant decrease in IgA targeting are enriched for transcription factors known to regulate filamentous growth, but more specifically are known to promote adhesion, biofilm formation and adhesion. And so one of the sort of main features that makes Candida albicans so notorious compared to Saccharomyces cerevisiae is its adhesion potential. And so Candida is so adhesive because it expresses large families of cell surface adhesin proteins that are enriched on hyphae and facilitate the adherence and invasion of host tissue. And Saccharomyces cerevisiae lacks orthologs to most of these adhesins. And so we asked whether these adhesins are directly targeted by IgA responses by leveraging a collection of Saccharomyces strains heterologously expressing different Candida adhesins on their cell surface. We then took all of these fungal strains, incubated them with intestinal IgA from mice that had been monocolonized with Candida albicans, and quantified IgA binding by flow cytometry. And we found that where Saccharomyces cerevisiae is alone, the IgA does not cross-react with Saccharomyces cerevisiae alone, but that expression of ALS1, ALS3, and HWP1 was sufficient to promote IgA binding, demonstrating that these adhesins are directly targeted by IgA within the gut. And so just a quick summary of what I've shown so far is that Candida albicans is a potent inducer of adaptive immune responses within the gut. And these immune responses target Candida hyphae and the adhesins enriched on those hyphae. And this is associated with the reduction of hyphae within the gut and selection for yeast cells. And so this is all very fascinating, but who cares? How does this actually impact both the fungus and the host? How does it impact their fitness? And so to first ask whether how this immune sculpting impacts Candida fungal fitness within the gut, I designed a conditioning and competition experiment. And so this involved monocolonizing either wild type B6 or reg immune deficient mice with isogenic Candida strains differing only by the fluorescent marker they express. I allowed these Candida populations to condition in these mice for either 2 days or 4 weeks, and then I mixed the fecal Candida populations together and competed them in a naive new set of mice and asked which population of Candida won. And so what we observed was something quite interesting, and that's that the Candida that had been conditioned in the wild type B6 mice had a significant competitive advantage over the Candida that had been conditioned in the immune deficient reg mice. I found that this required time of conditioning, and that if you condition the strains in mice for just 2 days, which remember is not a sufficient amount of time for adaptive immune responses to develop, I saw no impact on competitive fitness. And so this together suggested that Candida albicans actually benefits from interaction with the adaptive immune response. And in data I don't have time to show you today, we found that this immune enhanced fitness requires the yeast to hyphal transition. So there's been a number of labs now, including the Noble lab, the Kumamoto lab, the Pavelka lab, that have all demonstrated that the gut is a somewhat unique niche within the body in which Candida yeast have a significant competitive advantage over the hyphae. And our current model is that immune selection for yeast over hyphae makes the Candida population more fit for gut colonization. But of course, does this impact the health of their host? And so specifically, how does this immune sculpting impact inflammatory bowel disease? So there have been a number of studies now that have demonstrated that Candida albicans is potentially pathogenic during IBD in both humans and in mouse models. And the mouse model that we use for inflammatory bowel disease is this DSS model of colitis. And so this is a very simple model in which we simply give mice this DSS detergent-like molecule in their water, which goes to the gut, disrupts the intestinal epithelial barrier, and leads to pathogenic inflammation. And we know that if we combine DSS treatment with Candida albicans treatment, that disease gets even worse. And you can see this quantified here by scoring of colon damage from histology, that Candida treated mice have worse colon damage than the mice not treated with Candida, the mock treated. And so using this model, I asked how Candida morphology impacts disease using strain that I could lock in either a yeast form or a hyphal form. And using these yeast and hyphal lock strains, I found that the hyphal lock strain induced significantly worse disease than the yeast lock strain, suggesting that this hyphal lock strain in the context of inflammatory bowel disease may be the more pathogenic cell form. We also asked what impact adhesins specifically have on this exacerbation of colitis. And we focused on a mutant strain that was actually the sort of top hit from our genetic screen, this AHR1 mutant. And so AHR1 encodes a transcription factor that promotes the expression of cell surface adhesins. And so using this mutant, we asked whether this mutation also impacted colitis severity in this DSS model. And indeed, we found that this AHR1 knockout induced significantly less disease than the wild type Candida. And we asked whether rescued expression of a single adhesin that we found to be deficient in this AHR1 mutant could rescue disease. And so we found we overexpressed or constitutively expressed the ALS1 adhesin, which remember was a IgA targeted adhesin that we identified from our Saccharomyces strains. We found that overexpression of this ALS1 adhesin rescued disease to this AHR1 mutant. And this disease severity required ALS1 expression, because if we knocked it down again, it was indistinguishable from AHR1. So together suggesting that Hyphae and the adhesins expressed on Hyphae are pathogenic in this mouse model of colitis. But we wanted to determine whether the immune responses that target adhesins is actually beneficial in this context. And so to test this, we leveraged this Candida vaccine that many of you in the audience are probably familiar with, which is called NDV3A. And so these experiments were done in collaboration with Ashraf Ibrahim and Shakti Singh, who provided the vaccine. And so this vaccine induces an immune response specifically against one Candida adhesin, that's ALS3. And ALS3 notably was one of the most highly targeted adhesins by IgA that we observed in our studies in the gut. And so we asked first whether NDV3A vaccination could induce intestinal fungal specific response that we can observe. And so to test this, we vaccinated germ-free mice with either the NDV3A vaccine or an alum control, and then monocolonized them with Candida albicans. And what we found was that immediately after colonization, we were able to observe significant IgA targeting of the intestinal Candida population within the NDV3A vaccinated mice, but not in the alum vaccinated mice. And that imaging flow cytometry revealed that this IgA response was restricted to Candida hyphae. So we thought this was a really useful model to determine whether hyphal-specific antibody responses, what impact they had on intestinal colitis. And so using the same vaccination strategy, we carried out a similar DSS experiment with and without Candida treatment. And we're excited to find that in mice vaccinated with NDV3A, they were protected from the Candida-associated intestinal damage that occurs during DSS colitis. you can see there's significantly worse disease in the alum-treated group compared to the vaccinated group, and that this response was dependent on Candida treatment because there was no difference in disease between the mock-treated alum and NDV3A groups. And so altogether, this demonstrates a really interesting mutually beneficial interaction between Candida commensals and adaptive immune responses, where adaptive immune responses are induced by Candida albicans. This sculpts the Candida fungal population towards a more fit cell type within the gut, improving Candida's fitness, and also benefits the host by downregulating adhesive molecules that we found to be pathogenic in mouse models of colitis. This further highlights the potential therapeutic benefit of vaccination in targeting commensal fungal populations to restore homeostasis and prevent pathogenic inflammation that can occur in people with IBD. And we're excited that this study was recently accepted for publication, and I'm excited to share the whole paper with the community in the near future. Okay, so I have many people to thank for this study. The the Round Lab has been fantastic, but I also want to call out just the fantastic fungal community in this setting. I came to the Round Lab without knowing how to do much Candida genetics work, and I've leaned heavily on the entire Candida field. Many within the field who've been very generous with their time and protocols to help us get Candida genetics up and running. And I'd like to thank my funding source, the Helen Hay Whitney Foundation. So thank you.
Thanks so much, Kyla. That was a beautiful talk. I have to apologize. We lost your host, Rachel, just before you were supposed to go on. So I just want to quickly read your introduction, letting you know, let everyone know who you are. So Kyla did her undergrad at Montana State University and her PhD with Andy Ospa at Duke University. She studied alkaline response signaling pathways in the human fungal pathogen Cryptococcus neoformans. And for her postdoc, she moved to the Round Lab, but she's just told you about the beautiful and impressive work that she's done. She is on the job market for a faculty position, and she hopes to continue her studies on commensal fungi. Jessica, do you want to start us off with questions?
Sure. And so the first question is from Lucas Kuzabowski, and he asked for Vikas, why is alpha predominant and no A mating type generated?
Yeah, that's a very interesting and important question, because if you look at people who are familiar with the ecology of Cryptococcus neoformans know that more than 95% isolates of Cryptococcus are alpha types, and only a few of them are A types. So, and this was interesting in our experiments that when we did experiments using the lab strains of MAT-A, there was a roughly equal proportion of the progeny from both alpha and A. But when we did this experiments with the natural isolates, both PT63 and IUM96, we had only one case where A progeny was generated, but rest everything else was alpha. Now, I don't have a direct answer to the question that why that happens. Our hypothesis at this point is that there is something goes wrong with A parents. Either they are not able to sporulate on their own, or they have some sort of defect which inhibits their development into a mature basidia on their own. So they always need a alpha partner in cases. And but we haven't been able to pinpoint what that region is. We have done some initial analysis, which basically was a dead end. And so we're kind of rethinking our approach in order to understand what could be the determining factor there. And I would say if we are able to find that out, it'll be a really cool thing to know.
Thanks a lot, because I was wondering if this has something to do with the sequence of events that happened around the clamp cell when the nuclei are moving around that process of segregation of the nuclei. Maybe there is something consistent about alpha nucleus to progress more, and that's why you mentioned something about one of the nuclei to be kicked out or neglected during that process. Maybe that's why, because the alpha is always consistently at the forefront of that shuffling, and so the alpha progresses to the subsequent basidium while the A nucleus is getting neglected. But why would that be? I'm not sure.
Yeah, so
it seems very interesting.
We don't really know why, but we are looking into that. It might be something that is a phenotype similar to what happens in known as spore killer in other fungi, but in this case, it might be more like a nuclear killer factor which comes from alpha and kills the A or something of that sort. But it's all theory at this point, and we'll hopefully know the answer sometimes.
And so Xiaoran Lin jumped in on that and said, could it be related to the enhanced ability of alpha mating type nuclei to continue hyphal growth?
Yeah, I mean, for sure, it could be. But again, the question is why, what is it that makes A not do the same thing? Because, and so it's a question at this point that have multiple facets onto it, multiple ways to look forward to, and there is no direct answer, to be honest.
All right, great. Thank you for the discussion. Liz, would you like to take a question for Kyla?
Yeah, sure. So Kyla, everyone's asked the questions that I want to ask, so I'm just going to read from the list here. Jessica starts off by asking, oh, sorry, now I've lost you. Where are you? Do you think that morphology per se has disease-causing effects, or that morphological changes are associated with cell surface changes? So if you had the yeast alone expressing these antigens, would that be enough to cause the colitis damage that you're seeing?
I think that's an excellent question, Jessica, and I'm glad you brought that up. I think that the morphological changes are the impact or the importance of the morphology is more an indication of the effectors that the cells express. So it's about the effectors that are enriched on the hyphae, not necessarily the hyphal formation. And so the AHR1 experiment that I did, those mutants actually don't have a morphological or decreased in hyphae during gut colonization. And so even in the context of a strain that can make hyphae but makes less adhesins, we see less disease. So I think it's about the effectors and not necessarily the morphology.
Yeah. So Joe has a kind of a follow-on question with that, which is that, do you see if there's an IgA response against these cell wall components that are the classic immunological triggers, right? Beta glucan, as well as chitin, or in addition to these proteins?
Yeah, so I haven't tested that specifically and directly, but I will say that I don't see a lot of cross-reactivity of the Candida-induced IgA to Saccharomyces, for example, which also has a number of those same cell wall components. But I don't know for sure if there is a lot of reactivity happening in vivo, but at least when you take the IgA and ask it to bind to fungi in vitro, it seems to be fairly specific. And so maybe, but I don't have any evidence to suggest that's happening a lot yet.
Just one follow-up there is, do you have a thought about Malasthesia in the gut, which is this other organism that's been associated with?
Totally. I love Malasthesia. I have not done any work on that. I know that the Underhill lab, David Underhill Labs has a beautiful study showing that Malasthesia is also able to exacerbate IBD. And I believe in their study they found that there was some antibody binding to Malasthesia. So I'm not trained to work on Malasthesia, but I would love to, do it in the future. I think it's fascinating for sure.
All right, Jessica, unless Joe wants to comment there. I think go ahead and pass the next question to Vikas.
Joe, did you have a comment?
I'm just very excited to hear Kyla excited about Malasthesia as well as Candida. And if there's anything we can do to help, just let us know. We'd be happy to send you some strains. And
Yeah, we should be in touch. That would be so fun.
great, fantastic, spectacular talk. Look
Yeah.
forward to seeing the paper.
Thank you.
Great. All right, Vikas, from Matt Blango. Is there a hypothesis as to how the genetic differences between the strains are sensed molecularly? And is the shuffled strain generally less fit?
So I'll answer the second question first. Chromosome shuffle strain is as good as the normal wild type strain. We have looked at it in both mitotic assays, different stress conditions, and it doesn't show any differences as compared to the wild type. And for how the genetic differences are sensed, even with the chromosomal rearrangements being there, we're not sure at this point, but that's something we have been testing. We are now doing some assays with other strains which have different genetic variability and with respect to the wild type strain, and see if there is any difference that pops up there. It probably is similar to what happens when you have a strain without any chromosome shuffling. But if there is anything different, we hope to find that out by doing these assays with multiple different strains at the same time.
That sort of feeds into my question, which is that I think this idea of the morphological consequences of mating, which is to say the spore formation, can be more important than the genome recombination under certain scenarios, is really fascinating. And so I was wondering how common this type of non-recombinant spore formation is, and does its rate increase with genetic differences within species strain? So if you get really divergent strains, you can imagine a scenario where it might be in their advantage to protect their own genome.
Yeah, so like I said, I think we are looking at it. We are even looking at across species to see if you cross a neoformans with neoformans, which have much more higher genetic divergence among those, whether that will change the platform and give rise to more of these non-recombinant progeny production in the cases. So all that is basically ongoing at this point, and hopefully in next few months or so, we'll have a better picture of what's happening. And one thing that I would like to mention is for one of the strains that I didn't talk about, IUM96, it does have slightly elevated level of these non-recombinant progeny. So for both BT63 and the lab strain K99, we have it about 1%, but IUM96 is about 2.5%. So there is probably some variability, and it might be a strain specific phenomena for sure. And we'll have to just wait and see how it comes.
Mm-hmm. Very cool. Liz, would you like to take another couple of questions for Kyla?
Yeah, sure. Got a few more here. So Mark Swiggerdahl has asked, does the IgA response depend on T cells? And he expands on that by saying the UMT mice, which are B cell deficient, had a strong phenotype with extensive hyphae. And he asked, did you test CD4 knockout mice to measure Hyphae and IgA?
Yeah, that's a great question, Mark. Great to hear from you. Mark's right. A lot of the IgA actually produced in the gut does not depend on T cells. It's actually quite interesting. But at least for Candida, the IgA that targets Candida, that is dependent on T cells. So if we look at IgA targeting we use TCR beta knockout mice, so they'll lack, CD4, CD8 T cells. They largely don't, there's not very much IgA targeting in those mice. We didn't look at morphology in those mice, but given that they, lack T cells, which then also impacts the IgA targeting, I would expect to also observe an increase in Hyphae in those, but we didn't directly look in that mouse model.
So Matthew Blangos says, wonderful talk. Which I think we can all agree with, but he's wondering if you've tried to carry any of this into human models, for example, testing aspects in patient cohorts.
Yeah, so as a part of those human samples that we did use, about half of them had IBD. We did not see a significant difference in antibody targeting of Candida between IBD and healthy individuals, suggesting that, that maybe differences in targeting. IgA targeting is not maybe involved in disease severity, but it's definitely something we're continuing to study. IgA deficiency is one of the most common immune deficiencies in the human population, and that is associated with inflammatory bowel disease. And so that is something we're hoping to pursue, is to look at seeing if we can see something similar happen in humans. Yeah, that's a great point.
So Jose Vargas Muniz has a really nice follow-on question from that, which is a little bit out of left field, which is, so he says, for other pathogens such as chlamydia, it has been shown that the GI tract mediates protective immunity in mucosal tissues. For example, the female reproductive tract in the case of chlamydia. So he asks, have you seen a similar IgA secretion or protection in other mucosal surfaces?
Ooh, great question, Jose. Also great to hear from you. That I have not looked at other mucosal surfaces. I did see that there is a serum response associated with Candida colonization. And this was actually the topic of a really nice Cell paper that just came out of Ilya and Ilyev's lab, in which they found that that serum response is actually protective against disseminated infection. And so, but no, I haven't looked in like the urogenital tract or the oral cavity or anything like that, which is interesting. It would be a difficult experiment to do, because of course, when you colonize the gut, you colonize everything else too. And so it would be difficult to say like the stuff in the gut is driving the response in the urogenital tract, for example. So that would take some very fancy experiments that I'd have to think about. Yeah.
Fantastic. Thanks so much. Pass back to you, Jessica.
All right, comment or a question from Mohammed Hashim Reza for Vikas, and that's very nice talk. In the chromosome shuffled strain, do you see the other nuclei being propagated in the branched hyphae, with one nucleus being inherited uniparentally and seen in basidiospores?
Yes, so unfortunately, we haven't been able to image a hyphae long enough that we see the branch point and then both the branches are. The problem is all these assays are done on the agar plate, and a large chunk of those hyphae are embedded within the agar. So we've been trying to figure out how to do that in long term. But one thing that I can tell you is that we have occasionally seen cases where we have branching happening, and one branch has, say, a red nucleus and the other branch has the green nucleus. But we have also seen cases where a branch starts and it has on one of the nucleus, but the remaining branch has both the parental nucleus in it. So it seems to be a more complicated process than what we initially had thought, that it is just that a simple branch point and then they two separate from each other. But there are other possible factors that come into picture, something that Lucas mentioned, which is clamp cells, which are responsible for the nuclear migration during hyphae. And those are the explorations that are still pending. And once we established a better microscopy assay, I think we can answer those questions. But right now it's a technical limitation.
And the second part of that question is, if the better germination potential of spores generated from chromosome shuffled strains aids in propagation of the organism in the long run.
That's the hypothesis. So I believe that's what probably happened. So right now, what we think is combined together, if you put sexual reproduction and pseudosexual reproduction together for a cell, it gives us all kinds of advantage. So if it doesn't come across a perfect mating partner, it can just go through pseudosexual, make more copies of itself, and then go ahead, find a suitable mating partner, which allows all the recombination and purging of harmful mutations. So in the long run, I would say if you put both these phenomena together, it's much more better for a pathogen like Cryptococcus than just having one of the cases. Because if a new, for example, if a new genotype or a new karyotype is generated in nature because of all kind of things that might go wrong, then it may not be able to survive if you just have sexual reproduction in there, but it will survive through pseudosexual, and then hopefully find a partner somewhere else which have gone through a similar process. So I think in long run, combination of the two is something that's very helpful for this organism, I would say.
Thank
Great.
you.
Yes, thank you. And so that's all the questions we have for Vikas. Liz, do you want to take the last questions for Kyla?
I think we've gotten all Kyla's questions as well, so I think we can call it a day there. Really nice talks to both of you. Thank you so
Yes.
much.
And thank you so much for having me.